Good morning, everyone. Particularly welcome to those of you who've joined us on Facebook and YouTube. I'm Dr. Karen Miller from the Australian Institute of Marine Science, and we're here on board the RV Falkor. And we've just started our very first dive here on Ashmore Reef with the ROV SuBastian. We're currently sitting at about 150 metres depth, and we're here today to study the Mesophotic reef zones on Ashmore Reef. So what we're doing here today, and our very first dive, is really just having a look around, understand what's down here on the reef. What we're going to do first up is run a transect about 300 metres long, staying at about 150 metres depth, and you're just going to slowly watch us travel along the side of the reef. From these transect data, what we're going to do when we get back home into the lab is start to be able to quantify the cover and all the different organisms that you can see in front of you now. So already we're starting to see lots of cool things. There's some sea urchins, some crinoids. We've seen a few little corals so far. Oh yeah, there's a whip coral down there in the bottom of the screen. And yeah, we're just really trying to get a good understanding of what's living down here in this zone.
Don't forget, if you see anything cool that y'all need to come back to, let us know so we can wait.
So this trip on the Falkor is a collaborative effort. We've got a bunch of researchers on board from a range of institutions, and we've all got lots of objectives of trying to learn more about this Mesophotic zone. So we've got collaborators from the Australian Institute of Marine Science, from the Western Australian Museum. We've got students from Curtin University and the University of WA, Western Australia. The great thing about a collaborative cruise like that is we've got lots of people with lots of different expertise, and so we're really going to maximize the amount of information that we can get out of this trip. The guys from the museum are going to be later on in the day collecting lots of samples to take back for identification in the museum. We're going to be collecting samples to take water samples to collect eDNA or environmental DNA, so we can see, you know, if we can pick up all the species on the reef from the water samples and see how that relates to what we see on the videos. One of the exciting things we're doing here is we're testing a new system for monitoring the mesophotic reefs using hyperspectral imagery. So we've got a hyperspectral camera attached to the ROV, and we're collecting imagery along that will be able to relate to the visual imagery as well, and see if we pick up different things. And we're also going to be collecting corals for looking at their ecophysiology, for identifying them, and also for looking at population genetics of the corals to understand how related they are to each other all around the reef. So yeah, lots of things going on, but yeah, at the moment we're really just trying to understand what's down here. Hey, so we've just noticed that Linda Anderson is joined us on the chat, and for those of you who are excited to know, Linda's daughter Chloe is one of the students on board here with us on the Falkor. So Chloe, say hi to your mum.
Hi, Mom. Thanks for coming.
Hi, Chloe's mom.
You're a celebrity around here now, Mom.
It's great to have you along with us on this journey. Yeah, we'll make sure Chloe keeps you updated as what we're doing and what she's up to, and we'll keep her under control as well. Hey, we've also just noticed that Jeremy Horowitz is online, and Jeremy, you'll be pleased to know that the very first second we put the sub on the bottom, we saw a black coral, and we might even be the one that you want us to collect for you. So we're going to keep an eye out for you, so keep watching.
Hey Luke, I'm just going to keep updating your waypoint as we go. We're doing this transect here. I don't know if you see all the waypoints up there, so I'll just keep pushing it.
Great, thanks, buddy.
You're welcome, buddy.
We're about a third of the way through the first transect now. You can see that there's, not a huge amount of life down here, but if we zoom in a little bit, you can see there's lots of critters. It's quite exciting.
So this next transect.
Yeah, that's
Okay, cool.
Did you want to go, if you look at my screen right here, I'm actually J-rods. Go J-rods up. So we got in transect here.
Yep, that's
Do you
right.
want to just fold straight up to here? And
Yeah.
then transect this way and zigzag that way?
Yep, that's
And then
perfect.
likewise up here, we'll just zoom straight up like that.
Yeah, provided the
Yeah.
currents are happy for us
Of course.
to go either way. Yeah,
Yeah.
yep,
Cool.
that's fine.
That works.
Perfect. We'll be a well-oiled machine by the end of this trip.
Okay, so I'll put begin 100 transect right here.
That's
and
right.
end over there. And then
Yeah.
Okay.
Doesn't matter which direction we do them in, it's fine.
Sounds good.
So some of you are probably wondering what's so exciting about Ashmore Reef in particular. We're here at Ashmore Reef because it's an Australian marine park. It's one of 58 marine parks in Commonwealth waters around Australia. And this voyage is being supported by Parks Australia. All the information that we collect is going to help those guys understand a lot more about the marine parks they're trying to manage. So it's a really exciting opportunity. Ashmore Marine Park is really one of the jewels in the crown, if you like, of the marine parks, because it's got this fantastic coral reef as part of the park. We know lots about the shallow parts of the reef. You know, Australian Institute of Marine Science and lots of other researchers in Australia have done heaps of work in the shallow areas, you know, where you can get in the water and scuba dive and swim around and see the corals and the fish and all the marine life. But we don't know much about what's beyond those depths in what we're calling the mesophotic zone. And so that's what we're here to do for this trip, is to really understand the deep parts of the reef, understand the biodiversity, understand what's there, what's so important about it, map some of the areas, collect lots of samples. So it's going to be a real journey of adventure and excitement for all of us.
I want to pay out eternal.
Just 30 meters or so.
Feels like it's almost when he started moving.
Thanks.
Okay.
So this transect that we're running is the deepest that we're going to run today. And so that's probably why it's got the, you know, not necessarily a huge amount of stuff down there. It's going to be the lowest light levels. So we're sort of not seeing any of the organisms like the Spharectinian corals that like light and have symbiotic zooxanthellae. But yeah, we'll go up shallower. Our next transect's going to be up at 100 meters, and then our last one will be up at 50 meters. And by the time we get up to 50 meters, I reckon we're going to see some pretty spectacular coral reefs up there. So Nooney's just asked a question about whether we're going to be doing any 400 plus meter dives, and the answer to that is no. The reason we're not going that deep this time around is because we're really focusing on the mesophotic zone. Now, mesophotic, many of you probably know, translates literally as middle light, and so we're focusing on the areas that are in this middle light zone, so not the really bright areas that are the shallow reef, but basically the depths from around about 40-50 meters and down to around about 150 meters, which is about the extent that the visual light, the sunlight, penetrates down through the ocean, and that the organisms living on the seafloor actually need that light to survive. So we're really just focusing on that mesophotic zone for this trip and not going any deeper. The reason we're focusing on the mesophotic zone is because it's really poorly understood, particularly in this part of Australia. We know there's huge extents of mesophotic reefs across the northwest shelf, but because we've never really had the ROV technology like SuBastian available for a lot of research in Australian waters, we really don't know very much about them. We know lots about the broad distribution patterns, where the reefs are, and sort of the larger scale patterns of where species are around the reef, but we've never been able to get in at this really detailed scale before. And so that's what's really special about this trip, really being able to see all the individual corals and critters on the seafloor. So just past the halfway mark on this transect now, just going slowly. Just thought I'd take the time to tell you a little bit about what I've been doing here and my how I ended up here on the Falkor. My background is actually as a coral biologist. I started working with my PhD on the Great Barrier Reef on shallow water corals, where I started to try and understand the taxonomy of corals, their distributions and how they all interacted with each other. My career has been pretty amazing from working in the tropics of Australia. I've gradually travelled further and further south. I worked in New Zealand and I studied black corals in New Zealand and stylosterid corals over there as well. I worked on deep sea corals around Tasmanian seamounts. I've even studied corals in Antarctica, so I've pretty much covered the full spectrum of tropical to Antarctic corals and temperate ones in between. Decided Antarctica was a little bit on the cold side for me though, and so I've really enjoyed coming back up into the tropics and having the opportunity to sort of translate some of my deep water experience into some of the slightly deeper areas of the Northwest Shelf, so in the Mesophotic zone. I'm currently working with the Australian Institute of Marine Science. I've worked with them now for almost 7 years. It's been a great experience. I started out there as a research scientist, and yeah, my work was focusing on a lot of exploratory work, trying to understand the distributions of some of the habitats around the Northwest Shelf for coral reefs. Working with museum and other collaborators on trying to, look at where all the sponges were, provide information to inform marine parks and management, and also particularly to inform sustainable development along the Northwest Shelf. Through 7 years of working at AIMS, I'm now actually the research program director based in our Perth office. And with that comes lots of responsibility, and they've let me out of the office to do some really cool science for the next few weeks, which I'm really happy about. It's nice to be back out at sea and looking at the critters on the seafloor again, rather than looking at the computer all the time. There's a nice little interesting rock just going past now. Some nice soft corals and crinoids growing on it. That's an interesting cuttle bone there on the bottom. Here beside me, at Mission Control, I've got Nerida Wilson from the Western Australian Museum. Now, some of you might know Nerida from one of the previous Falkor trips. Nerida, do you want to say hello to everyone? And
Hey everyone, I know there's people from the museum watching and people from our previous cruise watching. About this time last year, many of you were along for the ride as we explored a couple of canyons near the Ningaloo area, which is really exciting, but I'm super pleased to be back here again, almost exactly 1 year later, and looking, obviously at slightly shallower depths this time, but still keen and interested to look at these reef habitats that we don't know very much about.
So are you seeing everything you expected to see at 150 on the transect?
pretty much. I mean, we're pretty close to the edge of the photic zone here, and so we wouldn't expect to see a lot of cover down here. I've actually seen quite a lot of this sort of part of the reef before, but the way we've always sampled it before has been with a towed video system, which means we've been running a towed video at about 1.5 knots across these sorts of habitats, which means you get a really good understanding of the sort of broad scale. Yep, we can see it's sort of a consolidated rubbly limestone habitat. We can see that there's the odd sponge or the odd coral here and there and the odd urchin, but we've never been able to see it at this level of detail before. So that's what's really exciting about this trip.
What are you expecting to see at 100 that you're not seeing here? Like a significant amount more, or?
I reckon we'll probably start to see a lot more big tall Gorgonian fans, a little bit more encrusting coral coming through. Still probably not a huge amount of cover. It'll get most exciting when we get up to the shallow bit, where we expect to see more corals. I'm a little bit biased because I do corals, and so corals is what I'm going to get excited about. But obviously there's going to be lots of other critters there, like sponges and echinoderms, and the odd mollusk, probably the odd nudibranch for Nerida. But corals rule the world, we know that.
Hot coral. Probably the center of screen.
I think that's probably
Yeah, I don't think it's a hard coral. We've got a team of helpers in the background who are trying to find all the really interesting things, because I'm kind of half looking at the screen and half looking at the chat stream and kind of talking to you guys, and I'm going to miss all the interesting stuff. So yeah, we're actually taking waypoints of really interesting critters that we see as we're going along, and we're going to come back this afternoon on our second dive and actually try and take samples of some of the interesting critters and take up close-up photos and get even more detail about the biodiversity that's down here.
The current has like I'm almost at about 64% on my lab.
Nooney's just asked if we're going to be sampling any fish. Nera, what are we going to tell her? No, not on this trip, I'm afraid. Or I'm not afraid. We're really focusing on the benthic invertebrates here. No fish this time around, but we'll be certainly collecting lots of pictures of fish if we see them, and they'll probably go back to the museum for the fish guys at the museum to have a look at, but not collecting any samples this time around.
What about the mollusks?
We certainly won't collect any baler shells.
Squid?
Will we collect squid? That's a question for Nerida.
Kitchen brush of science?
No, we did. Well, I think we might have packed one, but it's not. We don't really have that long mid-water commute. We are on the lookout for a couple of special mollusks that are found, we think, only at Ashmore. So we have a few targets in mind, but we're also interested in just learning generally about the community that lives here.
So we're just coming to the end of this transect now, just a few more meters to run. And then we're going to scoot up to 100 meters. And when I say scoot up, that's actually going to be move quite slowly up to 100 meters, the guys tell me. Yeah. And yeah, it'll be exciting to see what we find up there. And alongside me now, I've got Amy, one of our wonderful honours students. She's from Curtin University, and she's going to shout out to somebody.
Hi everyone, thanks for joining us. I'd just like to give a shout out to Kurt Crispin. He's a marine and estuarine ecologist from Perth and also my beloved partner. So hi Kurt, thanks for watching.
Amy, do you want to tell us a bit about what you're doing on this trip?
Okay, so I'm going to be looking at the ecophysiology of the corals. So hopefully going to be seeing whether any of our corals are in reproductive stages or reproducing, also where they're getting their energy from, so if they're feeding through their symbiotic algae or if they are heterotrophically feeding, and also what their algal density is as well.
So we've just been having a little bit of a poke around at the end of that first transect, and now we're heading off up to the next transect, which is going to be at 100 metres. Lots of action here in mission control as the ROV guys get their systems sorted.
What are you faced into the cliff and just lateral? So, were you down there watching? Okay, so this, let me get the camera back. So about, I was just pretty much locking in the depth at 150. You will be 100. Well, no, it gets about that close right there. Just about that close. I'm trying to keep this image.
1.5 meters
Ish, yeah, depending on that, you know.
and 90 on it for sure.
Yeah. Well, you can cut off, but as soon as you start cutting off, you'll start getting the shadows.
Yeah.
So it'll start getting blue on you. You'll probably have to do another white balance when you get up there.
Okay.
We had to do a lot just getting down here. So right now it's just going up, opportunistic, looking, whatnot.
Okay.
And then once you start to transect, it's just, I was going about 0.15. 0.2 was a little bit too fast, and 0.1 was okay. Anything 0.10 to 0.15 on that. I don't know if you use that bullseye right there.
Usually use about that.
Okay, yeah, so I normally use that one just because.
So 0.15 on that one.
Yeah, that's what I've been going by, and they were happy with it. So.
Okay, and then just opportunistic up to 100 and then 100 over.
Yeah, and
I'll leave
it's
you in
really
charge
not
of
even,
Mike, okay?
just you get a filter when you're up there, and then
Okay, cool.
when if you're still on after you finish that one, and you go this way, your umbilical's going to want to do a crazy kind of loop because of the current.
Yeah, just going to be kicked over on the right side there.
Yeah.
I wonder as we're coming up, I think Ben's on the back deck, but
Yeah,
just let him know that the floats might pop up.
Yeah, whenever you hit, I'm imagining 150 on pay-in,
Yeah.
then we really need to have somebody back there.
150 on pay in.
Yeah, because
what
we
about
don't,
208? Yeah, okay.
we don't want the floats to go over to the Dunkin' House.
Yeah, of course.
You might have to keep them a
If
little
you could
bit
just
further
let him
away.
know when you're out there. I think he's in the workshop. I didn't even tell him. I didn't know where he does work.
Roger that.
Awesome.
All yours start with the next transect, so I can start with the photos. That'd be great. Yeah. Just slight like soup kind Yeah. I might go get lunch. Do you want me to? I'm happy to keep taking still pictures with these in between bits. What do we want?
I mean, it is already taking.
So it's taking video and capture, so the different things. But if you're happy with the capture, that's fine.
Well, this isn't on a transit, so.
Yeah.
No.
Okay, no, I'm going to get lunch.
Yeah, I reckon I can just text you when it's time to start.
Yeah, we'll just walk next way.
Oh, yeah, okay, cool.
You can kind of.
We won't start without you. We can't start without you.
Bridge RV. Yes, we're going to be heading up to the next beginning 100 point, so I'm just going to move you to the north.
Okay, great. Just informed Eric of the plan, and he'll be taking over the launch. We'll be on comms for the next 4 hours.
All right, wonderful.
Great.
Very time off.
Cheers. Thanks.
Never down. The average.
So you might see that we've just placed a little overlay to show you where we are in the world. And so while we're in Australian waters, we're actually like a lot closer to Indonesia. You can just, in the bottom of that little corner, you can see the tiny bit of Australia peeping through, but it's not the main part that you're seeing. But Ashmore Reef is situated at the very edge of the continental shelf, but part of the Australian EEZ. So that map will help you kind of place yourself in the world while we're staring under the water at around, at the moment, 130 meters as we just move up to the second of the transects we'll be doing today, 100-meter transect. And, you know, if we turn the audio on and off, just forgive us. We're often swapping out people or concentrating on getting some of our science objectives done. So I know it's hard for you to judge if your audio is working, but throw us a bone. We've got a few things going on sometimes, so we'll chat to you as much as we can. So we're still just moving up to the beginning of our 100-meter transect. There's a frilly thing that you can see in the middle which looks like a nudibranch egg mass. So we'll keep our eyes out to see if the layer of that egg mass is anywhere around. You can see that the community's changed quite a lot from where we were at 150 metres. There's lots of different species that we're seeing now already.
Circle right there. Just look this. You should have any time if the surface is the same as down here. So separate right there.
Bridge ROV.
Bridge.
Hello, Eric. Whenever you go to that waypoint right there, and then we're gonna set up and see how that is for distance and do a white balance, and then probably start the transect to the east. So good.
Yeah, why not? Let's try it out.
All right. So yeah, just go for the next one, and then we'll play from there. Next
Yeah,
one.
alright, thanks.
Like, maybe when we get up there, we'll do that at 7 just to keep us on watch. Or 5-meter. I don't know what's, if we come up 50, we're at 185, we're gonna be close to 150, it should pop up on the surface.
Well, they're probably watching. I think
That is the, no. That's not the it's the green cable light line to the now.
Okay. Yeah, there's the alright, cool. Maybe a little uncomfortable.
Do me a favor there, just zoom me into 100. Just what that waypoint's like.
Leveling out pretty good.
Yeah, makes sense. Stretch it out here.
Maybe some.
No. Should come up here. Just get away from the ship because these pop up. Well, it's much further up to go. It'll be underneath.
Keep it further settled in.
Let's just see what it feels like when we get up there.
just to put every time I take 666, I just hear the noise that James makes. You know what I'm talking about? Yeah. It plays in my head. That's your lanyard. What? That's your lanyard. Oh, lanyard. It would be tied around the base of your. Perfect. That's what it is. You put a lanyard on your camera. Something to tie that is. That's tied to that. Camera on this. Yeah, Ben, you got me?
Roger that. Quick.
How are you looking out there?
It's hot. Milk's in the water.
Okay.
No floats on surface?
No floats on the surface.
Coming up 165, so you should be alright.
Roger.
Thanks, buddy.
Time to get Ian, I guess.
Yeah. Their temperature changing.
Let's just look at two of those.
So for this expedition, we're trying to understand the mesophotic reefs around Ashmore Reef more thoroughly. And what we're going to be doing is going to 7 different locations, and then at each of those locations we're going to be running video transects among other things, but we'll be, at each of the sites we'll be running one at 150 metres, one at 100 metres and one at 50 metres. So earlier we completed the 150 metre transect, and now we're just at the start of the 100 metre transect and just getting ourselves organised to begin that exercise.
I'll sit down with the white bells.
Grab some lunch.
Yeah, that'd be great.
Oh, thank you.
That is me. Okay, that's me sitting down. Alright, I'll just
Zoom in that sand right by the
yeah. That's stable. Let's just see. I just want to see what the camera is. What angle did you have the camera at approximately? Do you remember?
You would have to ask somebody with better knowledge than me about that one.
Okay, I just wondered
Well,
if you had a certain
So
angle for the
yeah,
transect.
on the last transect, it was the slope was really steep,
Right.
and so we had it basically facing straight out, but depends what the slope looks like here.
Are you happy with this kind of view here? I can come down a little bit. This is what's down.
Yeah,
We're setting up.
that's good.
So
I'll come up just to get that out of the way. There you go. Something like
there it goes.
that.
Yep, that looks good to me.
Okay.
Yeah, they look like the sand.
Hey, Chris, let me know when you get to 150.
Roger. Realistic sand.
Okay. And you are straight on
The bridge ROV, how's that feel up there?
Yeah.
Okay.
Easy control?
Perfect.
Yeah, we're nice and stable up here.
Alright, cool. Thank you.
Okay. You ready to start the transect? Happy bite.
Alright, let's start going.
Do him going fairly slow because I'm going to go fairly slow here. Alright.
Alright, that's us moving east, so we'll start you at about 1-2. See how it looks.
Alright, moving east.
Area for 2 seconds. Alright, we'll start right now.
So we're just starting the second transect at this location here on the southern edge of Ashmore Reef. We're at 100 meters depth. As you can see already, for those of you that watched the first transect, there's a lot more life up here in the slightly shallower waters. Really different reef substrate. Further deeper down, it was sort of consolidated limestone rubble. Here we see a lot more sand, but lots of soft corals, sponges, sea whips, a lot more diversity of animals at this depth and a lot more abundance of them as well. So this is going to be an exciting dive.
Zero's doing 0.1, 0.15, 1.5. So, definitely have the current on this one. Just slow it down.
We're running with the current this time?
Yeah, I actually keep kind of pushing back a little and just throwing me this way.
Yeah, okay. Water's nice and clear though, isn't it?
Very. We actually, we came up between, what was it, 112. You could see the shimmering of the water from just
Okay. Have we got the water temperature up somewhere on the screen?
he's not running there.
Ah, okay. No.
Yeah, you can definitely see we went to the thermal climb. Right?
Okay.
Yeah, we changed.
I missed that. I was having my lunch. Next time.
You're gonna miss it. Lunch is a good reason why.
Ah, nice sponge there. Beautiful.
haven't started this transect yet.
Oh, sorry, yes, we have. We've just started, so yeah.
Yeah. No, it's pretty good. I was worried it was going to start porpoising a little bit or something, but it seems to be.
Chloe, did you get that message from Merida?
Yeah. Oh, good.
Do we need to let your mum know that you are going to have lunch?
She thinks I'm starving. She knows we're eating well here.
Yep, they do look after us well here on Falkor, don't they?
Yeah, she was looking because she had egg on toast for dinner last night, and I was like, we had spaghetti.
Oh, there's a fish in that.
What was
Looked
that
like it
thing
got a bit of
underneath us
splash.
that looked like a kind of orange fanny thing on the Karen?
That gorgonian.
That big boy?
Oh,
Just like
what?
that.
Whoa,
We
whoa,
saw-
oh.
Yeah, we've seen some of that. I saw
So the big
What are those?
orange Gorganian fan.
Yeah.
Yes. Yeah, we're seeing quite a lot of those, aren't we?
Right. Get the camera in this way. So tough.
The substrate's quite sandy here.
Yeah.
You would have pulled the under. Okay.
They're on that like rocky limestone.
Yeah.
No, let him, because he's going a little bit faster than me. So let him get to that waypoint, and then I'll pass him, and then we'll belly hopping in. He's doing 0.5 on 0.2.
I can tell the kids through.
I think he's trying to make it right. So I'll first let him get set up to there, and then we'll pass me. He's probably going to put Kurt's pushing me up there.
Hey, could you guys get a point into the body, please? Looks like there might be some straight tunes. There. I just wanted to come back.
Are you there for the cave incident? Do you know?
Oh yes.
Oh, look at that thing.
Ooh, nice
Feel
starfish.
free, parent, to just yell out at me like POI, hard coral, blah blah, whatever.
Yeah.
I'm pretty
This
good
is
at the
dive one.
keyboard.
We're still getting kind of into the swing of it, aren't we? Yep.
Can you get that for me?
Current giving you grief?
It's just pushing us around a little bit, but are you, is it, you happy with
It's,
far?
yeah, it looks beautiful to me.
And obviously no stopping, you want to just continue right through.
Hey?
No stopping, you want me to
Yeah,
continue
just
right
we just want to keep going for this one. We're going to come back later this on the second dive. And
When you get to 130 now.
Roger, right now you're at 164 Ben Herm. We're pretty much staying at the same depth, so I wouldn't think it's going to change too much for the rest of this transect.
Roger that, thank you.
Yep.
It's actually kind of pushing me up into the
Yeah.
starboard there, so I think keep having to kind of slide back down to spread 100. Even though that's 97, you add the depth there to the bottom, so you're actually looking at 100-ish.
yeah,
99.5.
that's fine. Yeah.
to the mil.
Give or take a metre is not going to make that much difference. Are you happy with the temperature that's running at too?
Yeah, I'm just taking nice and slow.
Yeah. Yeah, so one of the challenges we've got operating in these relatively shallow depths with SuBastian is that we don't want it to get too warm, because the ROV's been built to operate down to thousands of metres where usually the water's quite cold. And here we're sitting in water temperatures of probably close to 30 degrees. And so we just got to be really careful that it doesn't overheat and we don't break it.
Over the past 8 months or so since we've been here in Australia, we've added a couple of cooling circuits to try to help that out. And I think it's made a pretty good difference. So
Fantastic.
It's helped out a lot.
For instructions to the jar can.
Very good.
Jar can.
Yeah.
You like that? I don't know why we were scared before. Maybe because the jars are cold.
Bridge ROV. I'm gonna keep you right here for just a second. I'll just get us a little bit further ahead so you don't have to take us, alright?
Alright, so stop mode here.
If you can slow down, you don't have to come to this stop. Just try to come and slow down if that's possible. I know current's pushing it.
will do.
Alright.
Oh, point of interest, guys. A few more of those corals by the looks of it.
Do you want us to mark those on ours as well?
That's a good question.
It makes it easier for us trying to get back.
Okay, yeah.
So if you saw
So
something,
if you
just throw away, and then we'll just say, marker 14 or whatever. Use
perfect.
Yeah. I mean, I can't be the easiest thing. If I run into crisis, I'm using it. Yeah. Yeah. Sure. Just throw it on the track there. Karen, I'm going to frame those halfway through again. And then the 50.
Yeah, we'll fire the Niscus halfway along this. So guys, can you let us know when we get about halfway along this transect?
Sure.
And then we want to fire the 3 Niscus models to collect the eDNA samples.
You're happy just to, we sit down to do that?
Yes, perfect.
Alright, so let's go right here. This point.
So,
So I've marked a few waypoints.
I did see an egg mass on Major at lunch that was a beautiful front egg mass. Same thing in
Oh, I hope you marked it. Come back now. There's another one.
Does anybody know how many meters across the field of view is?
That's another really good question. Do you guys know what our field of view is? Do you know what the field of view is?
On this Zoom?
I mean, that the 2 green dots are 10 centimetres, aren't they?
Yeah.
Yes.
Look at them all. Lots of them. Yeah, it doesn't sound on there actually.
From comparing that camera to that camera, it was about half the width of the ROV roughly.
Yeah, okay. And so that's, you know, from a science perspective, that's a nice size. So we've just reached about the halfway mark on this transect, and we're just going to settle down on the bottom, and we're going to take some water samples. So the water samples that we're going to collect are actually for environmental DNA studies, or eDNA. And what happens, we'll take the water sample here at the seabed, and when we get the sample up onto the boat, we'll filter it, and from what we filter out of the water, we'll actually then extract its DNA and sequence it and use that to try and understand how the DNA in the water column relates to the animals that are on the seafloor. So we're testing it as a tool to kind of understand and monitor the diversity on the reef. So if it turns out we can kind of identify most of the species that are on the reef just from the water sample, that's going to help us, you know, do lots of broad-scale sampling all over the place in the future. So guys,
That's pretty good there.
and Declan, who is one of our honours students on board from the University of Western Australia, is actually the person who's processing and collecting these samples. Declan, come and tell everyone about what you're going to do.
Alright, I think Karen's done a fantastic job explaining everything already, but yeah, so we're going to shoot off 3 of the Niskins now from the ROV, and then when they're up on the deck later, I'll be filtering them and trying to get all the DNA extracted from that water, and that water can just go into the sink, and we're interested in the DNA that's on the filter, and then I'm going to be rolling up that filter, putting it into a -80 freezer for storage back to the labs in Perth where we can process it there. Yep, so I'm very excited here on my first mission. Gonna hopefully get some really good samples.
This is your first ever time at sea too,
It is.
isn't it?
Yeah, got my sea legs now after a couple of days at sea. It was a bit touch and go in the first few days, but yeah, no, we're all clear now, feeling a lot better. The food here is great, so.
Yeah, you don't want to stay seasick for too long, do
No,
you?
no.
So I see, Justin, you've just asked about the new bio boxes. Very, very good spotting on that one. So yeah, under the ROV arm there you can see the new system the guys have developed specifically for this trip, because we want to actually collect lots of coral samples, particularly for population genetics. We needed the capacity to store lots of little samples rather than putting everything together in one big bio box.
That's one.
And so, yeah, so yeah, can you record the thanks narrator? So yeah, those new quivers that you can see down the bottom there with the numbers on them, that's for our population genetics samples. Whoop. And that's the second water sample. So we're about to head off along the transect again. Couldn't help but just have a little bit of a look around there at some of the amazing critters that were on that site. Huge amount of diversity, lots of sponges and corals, all sorts of really cool things.
Down here, so we're gonna start the transect again. So I'll get you in just 1 second.
Jump in a second. Alright, next.
Yeah, that's alright. Such a pleasure. Cool, thank you.
So we're back cruising along the transect at 100 meters depth. So we've just reached the end of our second transect, the one at 100 metres, which is quite exciting. Hope you enjoyed watching that one. Now we're actually going to take a little break for a minute because we've got some other operations on the vessel to do. So we're going to stop streaming the ROV for a little while. So good time for you wherever you are in the world to go and grab yourself something to eat. Stretch your legs, go and enjoy some sunshine or moonlight, and yeah, come back and join us in about an hour when we'll continue on with this ROV dive. So thanks for watching, and we'll look forward to seeing you again soon.